All right. Welcome, everyone. It's so good to see you all. My name is Shayna Salen. I am the program coordinator for the Osher Lifelong Learning Institute at Cal State Channel Islands. And it's our pleasure to be hosting the Power of Stem Cells, this wonderful stem cell seminar presented by Dr. Nitika Parmar and the cohort of MS Biotech students. So I'm going to go ahead and introduce Dr. Parmar now. Dr. Nitika Parmar received her doctorate in molecular biology from UCLA and completed a postdoctoral fellowship at UCLA. She received her BS in biophysics and her MS in biotechnology and MTech in biochemical engineering and biotechnology from the Indian Institute of Technology, New Delhi. Nitika's fields of interest include molecular biology, genetic engineering, and DNA recombinant technology. Her research focuses on understanding signal transduction pathways controlling cellular growth, normal and cancer, as well as studying protein damage in response to aging. She enjoys the excitement of research, but tremendously enjoys teaching and interacting with students as well. She feels that molecular biology has no limits and that its benefits can be tapped with an unending potential. The only requirement is being a sense of intense motivation and enthusiasm, which brings us all here together today. Before I pass it to Dr. Parmar, I just want to give a little plug for our summer taste session that's coming up. Dr. Parmar will be teaching a class with us in our summer taste session. Have 25 different one-time 2-hour lectures. Registration is in now. You can visit our website, go.ci.edu, and then click on the registration page. We also have the catalog there available for you. Classes start next week on Monday and run through July 17th. So we encourage you to sign up if you haven't already. All right, thank you so much. I will pass it to Dr.
Hi, good afternoon, everyone, and my special gratitude to all the students in the OLI class. You happen to be my favorite class. I've told this to Daniel and Shaina a number of times. It's a fantastic group. I love communicating and interacting with you. And thank you so much for joining this summer taste, which is actually going to focus on stem cells and how stem cells can actually be used to fix certain diseases and also in the area of regenerative medicine. So, Shaina has given a very gracious introduction to me, and I will not speak about myself, except that I have been at Channel Islands for almost 20 years, and I'm also a professor of biology in the biology program and also the director of the MS Biotech program. And it's my great pleasure and honor to have 12 of our MS Biotech students here with me, and they will also be presenting talks right after I have given my talk. So please bear with me for about 45 minutes, and then you'll get to hear some very cool stuff from 3 groups of students who incidentally will be starting their stem cell internship very shortly in about 2 months' time. And for those of you who are interested, we can let you know towards the end of the talk where all of these 12 individuals are going within California to conduct cutting edge science research in the area of stem cells. So what I'm going to do right now is I am going to share my slides. And if you have any questions, please, you could raise your hand while I'm giving the talk, or you could put it into the chat box. And I believe I have the honor of seeing as a moderator what questions are being posed, and I'll try my best to answer them. And this talk is simplified. It's fairly non-complicated in terms of scientific jargon. I know some of you may be, scientists who have retired, and some of you may not be in the science arena, as a result of which I have deliberately kept this talk more on the simplistic side here. All right, so I will jump right into it. So we do know the area of stem cells has become very hot for the past 5 to 10 years. And a lot is happening in the stem cell sciences area, particularly in California, Actually, California is leading the way amongst all the states in the US, trying to find cures for diseases, whether it's neurodegenerative disorders or whether it's different kinds of cancers, how stem cells can actually be used for a variety of reasons here. So these are a few Time magazine covers that came out, I think, 15 to 25 years ago. And this was a huge outcry at that point when people started reading the words cloning of humans and will there be another you and so forth. So the verbiage cloning actually was very exciting to the scientists, but it was very scary to the layman. So essentially, today also human cloning is not allowed, it is banned, but animal cloning is allowed. And cloning of genes is allowed to fix certain diseases in humans, particularly congenital diseases, which are tied to single gene disorders. So, what are stem cells? The title of my talk is To Use or Not to Use. Are stem cells actually a blessing, or are they going to be detrimental for us? And I will leave you with your opinion at the end of my talk. So the word stem simply means a very basic progenitor fundamental line from which others have arisen. And when I say line, we're talking about cells here. So in my talk, I'll be simply referring to human cells. As we all know, each one of us is made up of 3.2 trillion cells in our body, roughly. And that's a huge number of cells. So, how do we think all these cells are talking to each other? Some of the cells are dying, some of the cells are working very hard in our immune system. So, all these cells have different kinds of roles. Amongst this population of cells is a very tiny population of cells known as stem cells. And they're pretty tiny, actually, maybe only 1 to 2%. And these stem cells are playing an extremely important role in our body, although they are very small in number, but they play a hugely important role, which we will discuss in just a few minutes. So, what exactly is a stem cell? Stem cell is one of the coolest cells in our bodies, and it has two very specific features. The first one, it has a capacity to divide and make more of itself. And the second feature is it can specialize into any kind of cell in the body. So, a stem cell, which is classified as embryonic stem cell, can turn into any cell type in the body. It can turn into blood, it can turn into neuron, it can turn into muscle, and you name it, it can turn into anything, provided you give it the right food. And when I say the right food in biological language, we talk about proteins and what we call transcription factors, but basically these are proteins. So, depending on the cocktail of proteins that you feed to the basic stem cell, it will start differentiating into different cell types, which makes it a very important cell in the body. And of course, its feature to make more copies of itself
Put this in your
is also known as self renewal. And this feature is very important for day to day maintenance of the body. So you may not realize it, but within our body, the cells are getting old, the cells are getting damaged. Whenever we suffer an injury, the cells are dying. So we don't want to lose our healthy cells. So at that point, the stem cells come into play, the adult stem cells, and they start making more copies of themselves in a manner which will cause the body to undergo maintenance and repair and keep the body healthy. So stem cells, through these two features, is extremely critical that we understand that no other cell in the body has both of these features. A stem cell has the feature of self renewal, but the stem cell cannot differentiate further. It's already different. I'm sorry, a skin cell can divide extensively because our skin is exposed to the environment, and so we are always shedding skin cells. But the skin cell is already differentiated, so it has only one feature, self renewal. Likewise, a neuron, which is a cell in the brain, is highly specialized and differentiated, but it cannot make more copies of itself. So our adult cells in the body, majority of them can either do one, either making copies of themselves, or two, specializing, but they can't do both. And only stem cell has the capacity to do both of these features. That is why it is a gold mine in our bodies here. Right, let's move on. So self-renewal simply means that cells which are unspecialized, that means they are not having a specific functions, can make more copies of themselves and they can differentiate later. But right now they're reproducing themselves. And another term we refer to is unlimited potency. So potency means what kind of power does it have? And imagine somebody telling you you have unlimited power. I mean, that's an amazing statement, right? If someone tells me you have unlimited power to do anything you want to, that'll make me absolutely glorious. And this is what the stem cells are doing. They have unlimited potency to turn into any cell type in the body. Particularly, we're talking about embryonic stem cells, not the adult stem cells here, which I will actually clarify a little bit down the line. So, the richest and the most valuable cells in the body are embryonic stem cells. And of course, they are present in the growing fetus once once a woman becomes pregnant, and the embryonic stem cells are present in a little ball of cells that we call blastocyst, which I will show you. So stem cells can grow into large number of cells depending on whether the need is for repair or maintenance, or it can differentiate and become specialized where it has very specific functions to perform. So now I want to introduce to you the term potency. We use this very commonly in our biological jargon. There are three kinds of potencies in a body. First, pluripotency, second, multipotency, and third, unipotency. And it's clear from the verbiage here. Uni means one, multi means several, and pluri means unlimited. So a pluripotent stem cell can differentiate and adopt any function in the body. It can become blood, it can become kidney, it can become liver, pancreas, it can become any cell type in the body. So this pluripotency feature is only present in the embryonic stem cells, not in any other cell type. The second kind of potency is multipotent, where they cannot assume any function in the body. They cannot differentiate into any cell type, but they can differentiate into what we call a specific family of cells. And the best example of multipotent stem cells are the blood cells. So the original progenitor blood cell can differentiate into a red blood cell, a white blood cell, platelet, eosinophils, basophils, mast cells, name it. So there's a whole family of blood cells that we are talking about. But that original blood cells cannot turn into a neuron or a muscle cell. It can simply turn into the blood cells within its family. So that's why it's called multipotent. And the unipotent is the most restrictive. That means it can turn only in its own type. It cannot turn into different kinds of organs. It can only turn into one type, and that is a skin stem cell, essentially, where it is designed to turn only into a skin cell here. So you can appreciate amongst all the three, the pluripotent stem cells are the most valuable to scientists because they allow us to feed these cells different kinds of recipes of proteins. And then we wait a few days and then we look at them under the microscope to see what kind of cells they have become. Have they turned into a muscle or neuron and so forth? So pluripotent stem cells is where the maximum amount of research is going on today here. So what are the various types of stem cells? Initially, I talked about potency, but there are various types of stem cells in our body, primarily divided into four types: the embryonic stem cells, commonly referred to as ESCs, the adult stem cells, also known as ASCs, the cord blood stem cells, which are present in the cord blood, that means in the placenta, which is joining the mother with the growing baby, and finally, the amniotic fluid derived stem cells. And these are basically a very small population of stem cells which are floating in the amniotic fluid, which is providing a cushion to the growing fetus in the womb of a pregnant woman. So the embryonic stem cells are present when a sperm fertilizes an egg. It leads to the formation of a zygote. And then the zygote divides from 1 cell to 2 to 4 to 8 to 16 and to 32, and then it grows. So this is a specific stage. And please don't worry about the scientific jargon here. I just want to show you where the stem cells are localized. This is a very specific stage in developmental biology, very early stages of the development of the fetus, known as blastocyst. And these orange cells that you see here are known as the inner cell mass, ICMs, while the blue cells are protective cells which are providing support, and they're known as trophoblast. So scientists are interested in these red cells. Why? Because these are the embryonic stem cells. So if you were to question me, how do we isolate these embryonic stem cells for research? The answer is very simple. It will require destruction of the fetus because you'll have to isolate the blastocyst. You'll have to go with a very thin needle having an aspirator, and you'll have to suck out these cells from this ball of cells. So yes, there is a huge ethical dilemma when embryonic stem cells were worked upon several, maybe 18 or 20 years ago. Then the leftover fetuses were actually required to be destroyed, or fetuses which were discarded from in vitro fertilization trials were also given to the scientists, but no deliberate destruction of embryos was done at that point. But now we have come a long way, and science has advanced so far that we don't even touch the fetuses anymore, because we have actually created a ball of cells in the lab, primarily by a scientist known as Yamanaka, who actually got a Nobel Prize for it. And now we can create embryonic-like cells in the lab, in a petri plate, and essentially not destroy the fetus under any condition. And maybe one of our students can talk about that later. It's a very, very technical process of taking the adult stem cells, or rather adult differentiated cells, back in time and converting them into embryonic-like structures, which have embryonic stem cells in them. So right now, I just want to make sure you understand, none of the scientists, hardly any scientists, work with embryonic stem cells, which are isolated from fetuses. Now we work with what we call iPSCs, known as induced pluripotent stem cells. And this is a very cool picture. It's one of my favorite pictures. And this is an electron micrograph, which is like several $1 million worth of machine, which is looking at extremely small, cellular structures here, an early blastocyst and a late blastocyst. And these are real images here, which show you where the stem cells are actually localized. They are right here, the inner cell mass. And of course, this is an image which shows you how an embryo is formed when a sperm fertilizes an egg. And within 5 to 7 days, the embryonic stem cells can be harvested from the growing embryo. Of course, the growing embryo has to be growing in the lab in a petri plate. It's a plastic dish. And then once these yellow cells are isolated, they can be divided into 10 different plates or 20 different plates. And we can feed them different kinds of foods. We can feed them protein A, protein B, protein C, and there they will start turning into different kinds of cells. That is why they are so valuable. But as I mentioned in my previous slide, we don't need to isolate embryonic stem cells, because now we have what we call induced pluripotent stem cells, which do exactly the same thing that embryonic stem cells are capable of doing. So what are some of the challenges? I made it sound very easy, but science is anything except easy. It is very, very difficult because it requires a lot of intricate work. We are working with molecular molecules, which we cannot even see, but we have lots of fantastic equipment to measure what these cells are doing. The big problem for the scientist is, how do we determine what kind of food embryonic stem cells need for me to turn them into any cell type in the body? I mean, we have so many different kinds of cells. How do I know what do I need to feed the embryonic stem cells so that I can get skin cells from them? So here is where years and years of research has taken place all over the world, where through hit and trial combinations, different kinds of proteins are put into the house where the stem cell is growing, and we let them eat those proteins, and then we watch those stem cells after a few days to see under the microscope what kind of cells they have turned into. So you can imagine how laborious this is. There are 22,000 different proteins in a human cell. And now I'm telling you, we have to do all kinds of combinations to see which condition would be the best to produce skin, blood, liver, and so forth. So that's where the challenges are. Even today, there are currently those challenges do exist. So now let's move on to the second kind of stem cells, adult stem cells, as is evident from the name. These are not isolated from embryos or fetuses. These are present in our adult body, and each one of us has it. So these are also undifferentiated. That means they're very old in time. They're not having any specific function, and they are localized in a very small pool among the differentiated cell in pretty much every tissue in the body. And their main goal is to be involved in tissue maintenance and tissue repair. And if they were not present in our bodies will start breaking down every single day because we are working, we are walking, we are talking, we are lifting things, we are sleeping. So we are constantly on the move. So obviously, there's going to be some wear and tear inside the body, which we're not able to see. And the older we are, the greater is the wear and tear. So our body is so designed that these adult stem cells are actually involved in tissue repair naturally, which is great. And they have been found in a large number of tissues. As I mentioned here, adult stem cells have been isolated in several locations. They're present in the brain, the liver, and this is a partial graph. They've also been found to be in the kidneys and other organs, which are not listed here, but pretty much everywhere. And it's really important that we have adult stem cells in the brain. Why? Because the neurons in the brain have reached a point of no return. That means they have become fully differentiated and they tend not to divide any further. So if somebody has traumatic brain injury and they lose some of their brain cells, then the adult stem cells, which are sitting in the brain, they kick in and they start to replenish the cells which have died or cells which have become damaged. So it's very important that we have these adult stem cells for regular maintenance. So as I mentioned earlier here, these tissue stem cells, when the need arises, naturally the body sends signals to them. So here we're not talking about hit and trial. Since they are inside the body, they get various kind of signals, what we call inflammation signals, and different proteins, what we call transcription factors. And once they receive these, there is a lot of communication, a lot of cooperative talking between different cell types. And eventually the result is they know what to turn into. Unlike us in the lab, we have to experiment with a lot of proteins. Inside the body, there is a lot of collaboration going on. And so once they receive the right signals, that means different proteins are acting on them, they will start differentiating into the cell type that the body most needs. All right. So the key questions that arise with regard to adult stem cells is some things which we don't know still. How many kinds exist? Initially, they were found to be existing only in 5 or 6 tissues, and recently they are found to be existing in more than 15 different tissues. So we still have to explore, are they present in each and every tissue type, or are they only present in certain tissue types? But so far, the research suggests they are present pretty much in every tissue type. Then the second question, which I think is extremely baffling and interesting to me personally, is, are they arising on their own through an autonomous generation, or are they leftover embryonic stem cells? What is their source? Like, who's giving rise to these adult stem cells? Are they arising on their own, or are they actually leftover embryonic stem cells? So that's something that a lot of research is still going on. Third is, are they normally very plastic? Plastic means stretching. In this case, not stretching. In this case, it means they can turn into any type of cells. Or are they are very mischievous and they're very moody, and they have to be challenged and forced into turn into specific types. And finally, another super important question is, what kind of proteins act on these cells so that they can function in the repair of injuries and disease within our body? So there could be some other questions which are not listed here, but this is where the research is going on. Particularly number 4, extensive research is going on in that area, trying to find out what causes these stem cells to start dividing? What is the signal coming from the injured tissue? So we're talking about proteins here. The third kind of stem cell is isolated from cord blood, and cord blood is basically the placenta, which is found to provide the nourishment to a growing embryo or a growing baby inside the womb of a mother. And the placenta, which is shown here, it's a pretty taut organ and it's filled with blood, and that blood is very, very rich in proteins. And that blood also has a very small population of stem cells. And this population of stem cells tend to be hematopoietic in nature, which means they can only turn into primarily new blood cells, although there have been a few reports where they could be triggered into turning into neuronal cells also. So the good part about these stem cells is if a couple has a child which has some kind of a gene defect, and they don't want the second child to have the you know further siblings to have the same defect, what people do is they preserve their placenta and cord blood stem cells, which are sort of untouched and kind of clean cells, and they freeze them. So if the need arises down the line, if a subsequent child has some kind of a disorder, they can quickly isolate these stem cells, have them turn and differentiate into something which the second baby or the third baby may need. And this is, I wouldn't say a common practice, but several women populations across the world are choosing to preserve their placenta. And of course, you have to pay a price for freezing your cord blood stem cells, depending on the medical facility which you're utilizing for freezing them down. And then finally, the fourth kind is the amniotic fluid stem cells. And these are the least in population. As the name suggests, they are floating in the fluid known as amniotic fluid, which coats the growing embryo and basically provides a cushion so the embryo is not undergoing any kind of a damage as a result of some trauma to the womb of the mother. And they're very small in number, almost about 0.5 to 1%. And they have been induced to differentiate into different cell types. But honestly, not very many people work with amniotic fluid derived stem cells. And the reason is to isolate these stem cells, you have to go with an aspirator and the needle, particularly at the point of amniocentesis, which I believe is in the third or the fourth month, where a few fetal cells are removed from the fetus to look for genetic disorders by looking at the chromosome structure and integrity. So if somebody has to isolate these amniotic fluid stem cells, they'll need to go with a needle, and the risk is too high. And if the needle actually touches the growing embryo, there is a very strong likelihood of the embryo suffering a permanent damage. And that is why people don't like to work with amniotic fluid derived stem cells unless it's an aborted child or unless the child has stopped growing within the womb, in which case the amniotic fluid could be utilized. And so this is basically how it happens. Like I told you, the baby is coated by amniotic fluid, pale yellow color, and the doctor has to go with a needle, and you want to make sure the needle does not touch the baby under any condition, simply removes the fluid which is surrounding the baby. Right, so now the big question that comes up all the time to me when I talk about stem cells is, as I mentioned earlier, the embryonic stem cells are the hot cells because they are pluripotent. So in the days gone by, like 10 or 12 years ago, or even 15 years ago, how did scientists get access to embryos? A pretty big question. Mostly these embryos were obtained from in vitro fertilization trials conducted at fertility clinics, where a couple chose to have 5 or 10 embryos in vitro fertilized, and they chose to have only 2 or 3 children. And the remaining embryos were left frozen. And now the couple says we don't want to have any more children. So what do you do with the leftover embryos which are sitting in the freezer? So those are the embryos, if the couple agrees that their embryos can be donated for scientific research. That's one way. Second is where the embryos are formed, but they're not put back into the woman's body for further growth because these embryos have genetic defects. And so the couple has decided we don't want this embryo, and that embryo can be utilized for scientific research. And those embryos are very, very valuable because we can look at what kind of problems the protein signals have that causes a genetic defect to that specific growing embryo. And thirdly, are developmental disorders, which may or may not be tied to genetic defects. And very early in developmental biology, it's a very complicated procedure. If the protein cooperative talking breaks down, they could be lack of gene expression or overexpression of genes, in which case the embryos can be malformed. That means they may have certain kind of developmental defects, could be neuronal, could be limbs, could be eyesight, could be anything. And so, since those embryos are not being implanted back into the mother, those embryos are also available for research. So that is primarily how embryonic stem cells were isolated about 20 years ago through leftover embryos coming from fertility clinics. So now the big question is, why don't we use adult stem cells? Why do we always, now this is going back 15 or 20 years ago, why do we always run after embryonic stem cells? Two reasons. One, the adult stem cells don't have the same potency like embryonic stem cells. As I mentioned earlier, embryonic stem cells can turn into any kind of cell type. So they are highly plastic, while the adult stem cells have very limited plasticity, and that is why they were not worked upon earlier. And second important factor is these adult stem cells do not have a very wide genetic diversity because they're adult. So they've become differentiated. While the embryonic stem cells have unlimited genetic diversity because they are yet to grow in different kinds of cell lineages that are going to be happening later here. All right, I am going to pose a question here. I'm actually reading the messages which are coming through. So Eileen has a question. What about using genetic material after a miscarriage? Products of conception? Yes. Yes, Eileen, the genetic material has to come from the cells. So we cannot reach for the DNA and RNA directly. We have to get the cells coming from a miscarriage, and those cells can be very useful to determine what is the reason for the miscarriage. So of course, you get the aborted embryo, you isolate the cells, you grow them in the lab, and then you focus on the chromosomes inside those cells, particularly the DNA. It's a pretty costly procedure, by the way. We do whole genome sequencing, which can run into thousands of dollars. But if somebody is very interested in to find out what led to that miscarriage, it's actually doable in the lab to determine what aberrations or problems happened at the very early genetic level which caused the miscarriage to occur. So thank you for that very important question. All right, let's continue. So there is always an ethical dilemma. When people said, when adult stem cells were not discovered, scientists only had embryonic stem cells to work with. This is like maybe 15 to 20 years ago. So the question is, there were parties on both sides of the fence. There were people who supported the use of embryos for isolation of embryonic stem cells, which means destroying the embryo and actually utilizing the hundreds and thousands of frozen embryos, which are sitting in fertility clinics and which would not be utilized under any condition by the couple. So the proponents say, what are you going to do with all these frozen embryos? At least use them for science so we can try to find out whether certain diseases can be fixed. So they are in favor of utilizing those embryos. Then the opposite side are the opponents who say, no, these should not be utilized because embryos are life. And if allowed to grow within the body of a woman, not in the lab, they can generate a whole human being. So you can see the ethical dilemma here. One side is don't destroy life. The other side is destroy life, but for a good reason, that it can generate data which will tell us how certain diseases can be fixed. And then opponents also say, even if leftover embryos are not going to be used by the couple in vitro fertilization trials, at least these embryos can be given up for adoption to couples who are infertile. So both sides of the party are correct in their own way. But in terms of science, if we don't have access to this, again, we would have some restrictions and limitations in conducting the research. But now we have completely bypassed that stage, and hardly anybody works with embryonic stem cells. So, what can we use? What can be done with all these human stem cells? I can make a huge list because they are so valuable. We can study what happens in the very early stages of developmental biology as the cells are dividing and growing and specializing in the body as well as outside the body in lab. We can also utilize the stem cells as model systems to do what we call a high throughput screen to discover new drugs which can aid in the stem cells getting differentiated. And thirdly, which is very important, we do need to work with these stem cells in the lab to come up with what we call cell-based therapies. Cell-based therapies means we are utilizing these stem cells to produce a population of differentiated cells which can be embedded in the body of a person who has some kind of a disease, such as Parkinson's, Alzheimer's, diabetes, and so forth. So cell-based therapies are actually pretty hot area right now, and a lot of companies have moved because if these cell-based therapies come in the market, by the way, they have come, some of them have already come out, particularly for arthritis, for replacing the damaged cartilage cells. But for others like neurodegenerative disorders, we still have quite a long way to go. All right, so if stem cells are being put back into the body for whatever reason, regenerative medicine, to fix any disease, these stem cells have to satisfy a large number of conditions. And I'm just going to break it down here. First, they should be able to divide. There's no point putting cells back into the body if they're not going to divide. That means they're just dying there. So one is they should be very healthy and they should be able to divide. Second is they should be able to generate sufficient quantities of tissue. Not that they divide like 20 times just to yield a very small piece of tissue. They should be able to generate a large quantity of tissue. Thirdly, they should be able to acquire the function of the cell that they are replacing. That means they should be able to differentiate. Fourthly, which can be very critical, is after transplantation, they should be able to survive in the area where they have been placed. If they're not able to survive, because our immune system can get aggravated and it may start attacking those stem cells. So that should not happen. They should be able to survive and establish themselves and basically become friends with all the cells that are surrounding these external stem cells. If they don't become friends, then they become enemies and the internal body cells will start attacking them, and that's the end of the transplant. And on the same lines, they should be able to integrate and become happy with the surrounding tissue. They should be able to perform their function very well for a long period of time. Not that they perform the function for 10 days and then they fizzle away. No, they need to be functional for a long period of time. And finally, of course, they should not harm the recipient in any way possible. So right now, within the US, these are just US numbers here. These are people within the US which are affected by diseases that can be alleviated or fixed down the line through the use of stem cells. And of course, the number one on this list is cardiovascular disease, where people suffer myocardial infarction, where certain cells in the heart die off. And unfortunately, those cells are dead. They're not going to be revived. They're gone. And the muscle is dying in the cell. So what if we were able to get cardiomyocytes, which can actually be transplanted into the heart and they can differentiate and form more muscle cells? I mean, that would be too good to be true. We have not reached that stage yet for a wide number of challenging reasons. Then they can be used for autoimmune diseases. They can be used for diabetes, osteoporosis, and a whole bunch of neuronal disorders, and particularly for burns also. Yes, for burns, they can be very valuable, where particularly for third-degree burns, where the skin is completely charred, these stem cells can be used to form new skin here. So a large option and a large potential for usage of stem cells. right. So this is coming from the ISSCR website, where they have stated, quote, in the case of embryonic stem cell research, the end that scientists hope to achieve is the relief of human suffering. That this is a humanitarian and worthy end is not in dispute. The controversy is about the means, namely the consumption of donated embryos. That's where the ethical dilemma comes into play. All right. So, are there any questions I can take before I move on? Let me see how much time I have. I have about 7 to 8 more minutes. Are there any questions? You can either put them in the text box, or you can probably speak up or raise your hand, whatever works best for you. Okay, if there's no questions, I will quickly move on talking about the second ethical dilemma, which is actually talking about cloning here. Cloning simply means, the word clone means making more copies of yourself, whether it's DNA or RNA, whether we are making thousands of copies of DNA through PCR. Cloning simply means making an identical copy of yourself. So cloning has been done for human cells, where somebody suffers a genetic defect and now they have a bad gene. So we get the good gene and we engineer that in the lab, and we put that good gene back into good cells, and those good cells are put back into the individual who has a genetic disorder. And now the good gene produces good proteins, and the good proteins take over and completely dominate over the bad protein. So I'm putting it in extremely simple language here. It's extremely complicated to do, by the way. So this entire process is known as somatic cell nuclear transfer, SCNT. So this kind of cloning, which is happening in human cells, is allowed only in somatic cells. And somatic cells are those cells which do not turn into either sperm or egg. That means these are non-reproductive in nature. So any engineering or bioengineering that is being done through somatic cell nuclear transfer stays with the individual and does not get passed down to the children, because these are in somatic cells. But somebody can argue, why don't we do reproductive cloning? That means the traits or the characteristics or the features can be passed down to the children here. And thankfully, reproductive cloning is prohibited at the international level and is completely banned. Although there have been reports where certain scientists have engaged in reproductive cloning to produce twins which have the same kind of features, same hair color, same eye color, and so forth. But technically, reproductive cloning is prohibited for various reasons that we won't have time to go into right now. But we cannot start modifying human beings and create them based on my shopping list. My shopping list would be, I want a child who is 6 feet tall, has blue eyes, black hair, skin very smooth, is going to be an athlete, so has to have very good muscle mass. So we are tailoring what we want in terms of a children, and that is not allowed. That is ethically a grave, grave problem. So reproductive cloning, thankfully, is prohibited. All right, let me look at few questions here. Yes, so Barb Pilleri has a question. Can stem cells used to cure an adult with severe hemophilia? And yes, Barb, my question is yes. And the answer is very simple. The hemophilia is happening as a result of a single gene defect. There is a single gene defect on a beta globin gene, and globin is present in RBCs. It's a very major protein. So when this happens, the globin gene or the globin protein, instead of looking like this, becomes a bit bent. It becomes like this. And so the red blood cells, rather than being very nicely concave, actually become distorted. And so they're not able to transport blood, and then the whole havoc breaks on. Yes, for single gene defects, one can cure hemophilia by isolating stem cells by doing somatic cell nuclear transfer using a healthy nucleus, which does not have that particular beta globin gene defect, allowing those cells to grow in the lab. And once we have a large population, putting it back into the person who has hemophilia, and then waiting. The waiting period is where we wait with bated breath to see whether they are producing the healthy, normal beta globin protein. So, yes, I would say for single gene defects where the aberration is because of one gene, it's much easier to do. And then second question is through David Lowe. Where are we in replacing damaged brain cells? Good question, David, because this is a multi-billion-dollar market, considering Alzheimer's, Parkinson's, Huntington's, multiple sclerosis. I would say, I don't want to sound pessimistic. There's a lot of good research going on, particularly in Stanford, as well as, I believe, Cedar Sinai. They focus extensively on neurodegenerative disorders. We we still have time to go. It's not going to be coming out of the market, in the next 2 or 5 years. There's a lot of challenges. How do we transplant these cells directly in the brain? That means you'll have to do a very invasive brain surgery and actually guided by a robot assisted surgery, so that these cells, the healthy cells, actually cross the skull and be embedded in the area, whether we're talking about neurons or glia or astrocytes or whatever specific brain cells we are talking about. They have to be placed in very specific positions. And brain is a very tight organ. It's extremely tight. There's hardly any place for cells to move. And we have the blood-brain barrier. And, it constantly needs blood supply. So we want to make sure that even if we were successful in transplanting these cells in the brain, they should receive adequate blood supply so that the cells don't die off. That means the angiogenesis has to be pretty robust in the area where these cells have been replaced. So I would say not too close, but I would also say not too far away. Third question is Walter Johnson. Do insurers fund stem cell treatment? That is a super exciting question. Insurers, medical insurance companies, actually, I don't have an answer for that. I'll be honest with you. I don't have an answer, but I can get an answer for you. This is something I've never looked into, whether a medical insurance company will fund stem cell research. I'm not sure. But Walter, we can look into it and get back to you. And then Julie has a question or a comment. Quote, a company has been selling stem cell injections to inject in joints instead of having a knee replacement, for example. They asked for $15,000 for this injection, and there are no guarantees other than they will give you a second injection in about a year and a half, which may or may not work. Hard to tell these days if they are selling snake oil. Yes, there are a lot of snake oil compositions being sold today, not just in the US, all over the world. In fact, people are capitalizing. I've heard of so many cases, and we actually talk about this in some of our classes where there are ethical dilemmas which are raised by individuals, that whom should I trust? $15,000 is a lot of money for one knee, and if you need a second injection, that's $30,000. So knee replacement is a tried and trusted method, and it works pretty much all the time. But it's pretty invasive and requires a long period for recovery. So if somebody says, I'm just going to give you a shot and you'll be out of my clinic in 5 minutes, well, somebody would take that shot, right? Not have to undergo surgery and so forth. But there are people who do it perfectly well. The stem cells are injected into the cavity in the knee, and over a period of several months, the lubrication is generated and the joints start healing. But there are also people who take you for a ride. So I would say it really depends on what kind of homework you've done. There are people who are out to make money, and there are people who are genuinely interested in helping you be able to walk again. So I think it's just a question of doing your research and trying to find out how scientifically valid are the credentials of the person who is making this claim, and how many patients have already benefited, and what kind of reviews are there. And today, people can post any kind of reviews, and we don't even know if those reviews are good. So one has to be pretty smart and through word of mouth, understand who is selling a snake oil and who is actually selling the real stuff. Julie has another question. Will artificial intelligence soon help make advancements in this field? Yes, absolutely. I would say without doubt, yes, a resounding yes. The company told us that insurance companies will not pay for these injections. Doctors won't ask for it. Okay, so we got the answer. Thank you so much for giving that answer. They told us that Tiger Woods received this treatment, and I am not following Tiger Woods, so I don't know if he received this treatment and what kind of what was the outcome of that treatment. Colleen Nevins has a comment. Hey, Colleen, nice to hear from you. The trend is that if not FDA approved or a treatment deemed experimental, insurance will not pay for stem cells. I believe coverage is very limited. And Colleen has been my colleague at CI. She's retired now and she was in the nursing program. So she is the perfect expert to let us know what kind of things are approved in terms of medical insurance companies and which are not. So the bottom line is if something is FDA approved, it goes through the whole rigor of testing and experimentation, and it shows efficacy and safety, then the medical insurance companies are likely to pay for it or approve it. But otherwise, as Colleen says, the coverage is extremely limited currently here. All right, great questions here. Any other questions? So I think it's 1:55 almost. So what I'm going to do is I'm going to stop here right now and we will take about a 4-minute break and then we'll be back and I will have very interesting talks presented by our 3 groups of stellar students in the MS Biotech program. So let's meet at 1:54. Let's meet at 1:59 and we'll have the students presenting at that point. Thank you so much for your attention. Hey, Kaya, can you please upload your presentation? We have about
Yeah, you
one.
want me to start right away?
Yeah, why don't you actually, let's give a minute. I told 1:59.
Hmm.
And then if you could kindly introduce yourself and the team members in just about a minute, and we can get started.
Yes, got it. Okay.
And if you can also, four of you, quickly mention where you're going to be interning, really quick.
All
Just
right.
a
Okay. Um,
nine. Go ahead, Kaya.
okay. My name is Kai Heath. I'm a part of the stem cell emphasis. I'm going to be going to UC Santa Barbara, where I'll be studying heart cells. They're being differentiated via stem cells. Today, I will be talking to you about how stem cells are being used in the treatment of leukemia. I'm joined by my classmates Chris, Naga, and Sandeep, who will also be presenting. Um, and I will let Chris take it away from here.
Hello, everyone. Pleasure to be here. My name is Christopher J. Torrio. I will be going to UC San Diego under Dr. Gleason's guidance and studying multiple neurological diseases and using stem cell to basically differentiate it into neural stem cells and study using those as precursor cells. And for leukemia, I will be presenting on how blood cells are made, the types of leukemia and what is leukemia, and therapies we have currently in today's sciences. So how are blood cells made? We have two processes on how blood cells are made, hematopoiesis and leukopoiesis. The difference being basically hema being the red blood cells as we see on the right, and leuko being for the white blood cells. However, the process is very similar. They're both made by the stem cells created in the bone marrow that differentiate into the formed elements of blood cells that you see on the right, along with other immune cell types. So what happens during leukemia? So as explained by Dr. Parmar, these stem cells are very powerful. These are very wonderful regenerative cells. However, they are not invincible. They have a potential to mutate, and once under mutation, it could create a devastating trickle-down effect where basically these cells, even though they differentiate, they become unstable. They proliferate, they become cancerous cells essentially, and they are arrested in the differentiation state. So they are not stable cells. So they're just infinitely multiplying. They're taking up nutrients. They're limiting the function of other cells. They're taking up extracellular space, which can damage the internal responses around the system. And there are, for these examples, four types, which is acute, chronic, lymphoblastic, and myeloid. The difference being for these being is that acute is a rapid onset that is seen in young children and older adults, and this is a more dangerous type because this is a very rapid progression of the cancer, while chronic is a slow onset. However, needless to say, both of them are completely dangerous for a human. And lymphoblastic is explaining more for our innate immunity cells, which are red blood cells, white blood cells, and some other forms of platelets and types of cells, and myeloid being basically our adaptive immune system, which is what you would hear as the immune system cells, which are like B cells, T cells, and other attacking cell types. So let's go over the standard treatments that are currently being organized for to treating leukemia. So we have chemotherapy, which is essentially what we all know as introducing toxins or chemicals within the body that would target the lump of mass of cancer cells to an attempt to stop it from growing further. We have targeted therapy, which utilizes actually monoclonal antibodies in order to target specific functions, proteins, expressions of the cancerous cells in order to stop or even inhibit their function. We have radiation therapy, which utilizes the same strategy, which is basically using the chemotherapy's approach of a deadly radiation treatment toward the lump of mass of cells in order to destroy it and limit the amount that is currently there, and immunotherapy, which is introducing a, a a mix of serums, drugs, etc., in order to build up the immune system of the body, which is already compromised due to leukemia. And what we will be going over, which is bone marrow transplant using stem cells.
All right. So, a stem cell transplant is the process of replacing the cancerous bone marrow within a patient with newer, healthy bone marrow. This is typically done via surgery where a donor has some of their bone marrow extracted and then inserted into the patient. This is often done alongside other treatments such as chemo or radiation therapy, which remove a lot of the bone marrow that's cancerous and then can be replaced by the stem cell or bone marrow transplants. For patients who are eligible to receive transplants, remission odds are greatly increased by this. So, as said before, stem cell transplants offer a significant increase in survival and remission odds for those with leukemia. For adults with acute myeloid leukemia, for example, their survival rate nearly triples from 24% to 65%. Currently, there are over 80 transplant centers across the country where more than 10,000 transplants occur each year. The National Marrow Donor Program, an organization that works to match donors with patients, currently has over 22 million potential donors within their database. So while stem cell slash bone marrow transplants are extremely helpful, there are some challenges and risks involved. The key reason the National Marrow Donor Program exists and has such a large database is due to how complex cell compatibility is for bone marrow. I'm sure most of you are familiar with blood cell types, because with the 8 standard types being A, B, AB, and O with positive or negative. But for most other cells in in our bodies, there are a lot more types or surface markers that are used to recognize if a cell is foreign or like a friend. Um, there are more than there are thousands of these combinations, and while these markers are inherited genetically, due to how many combinations are possible and you getting them both from your mom and your dad, the odds of finding a compatible match even within your immediate family is around 20%. Mismatching of these surface markers between the donor and host can cause graft versus host disease, which is where the donor and patient cells recognize each other as invaders and attack each other. Because the markers are genetic, it is you're most likely to find donors, matching donors of the same ethnicity as the patient. But this makes finding donors especially difficult for minorities as they have much less representation within the donor databases. And Sandeep will now take over for the current advances.
Hello everyone, my name is Sandeep. I'll be going to citizen in Dr. Richie Ho's lab by studying the late onset diseases using human iPSCs. And today I'll be walking you through some of the exciting recent advances in stem cell therapy for leukemia. Before we dive into details, let me give a quick overview of the key breakthroughs we are exploring. We now have Amudacel, which is a recently FDA approved cord blood therapy that speeds up immune recovery and reduces infection risk. Scientists at the Biotech Children's Research Institute have successfully developed the first lab-grown blood stem cells, which are capable of repopulating bone marrow in animal models. There are also growing evidence for microtransplantation, which uses HLA mismatched stem cells, and also have strong results in improving survival while reducing complications like rejection. And finally, there are programs like NMDP are using AI-driven donor matching and gentler pre-treatment regimes which helps more patients get transplants faster and safer. Now let's take a closer look at each of these breakthroughs, starting with cord blood transplant and computational therapy. Computational therapy, which is being approved by FDA in 2013, which is an enhanced cord blood stem cell therapy that has been clinically proven to speed up white blood cell recovery and reduce rejection risk in the patients under transplant. In phase 3 clinical trial, the patients receiving Omburacil reached neutrophil recovery in 12 days compared to 22 days using standard cord blood, cutting recovery time almost half. Which makes the cord blood even more valuable is that it is readily available and has a lower risk of graft versus host disease, which is GVHD. This is especially important for the patients for diverse ethnic backgrounds who often struggle to find matched donors in the traditional registries. Also, Umbrisil not only enhances recovery but also expands access to transplant therapy for more people. Now, while Umbrisil improves how we are using donated cord blood cell therapy, we'll get into more details for the creating of stem cells in the lab. At the Muirdick Children Research Institute in Australia, scientists successfully grown the first lab-grown hematopoietic stem cells that can closely mimic those found in human body. These lab-grown HSCs were able to repopulate bone marrow in animal models, which opens up exciting possibilities for personalized stem cell therapies, especially for children with leukemia who don't have a compatible donor. Alongside this, there is another innovative approach called microtransplantation that shows great promise in elderly patients with acute myeloid leukemia. This method uses HLA mismatched peripheral blood stem cells, so even without a perfect donor match, the cells are infused after chemotherapy. What a remarkable patient's improvement shown is the survival rate and very low incidence of GVHD, which makes it a less intense but effective option, who may not be eligible for full transplants as well. So while a lab-grown stem cells may be future, microtransplantation is already giving the new hope to the patients who were once considered too high risk for standard therapies. And to make these kinds of treatments more accessible, technology is now stepping in to streamline how we match the donors and patients. Naga will walk us through this.
Thank you, Sandeep. And hi, everyone. This is Achyut Reddy. I'll be going to Stanford to Dr. Hagi lab under the observation of Dr. Michael T. Longacre. And we'll be working on plastic and reconstructive surgery and also regenerative medicine. Yeah. I'll walk you through the NMDP advances in transplant support. NMDP is National Marrow Donor Program. And as we were talking about, as Julie asked a question, will the AI be making any advancements in this medical field? Yeah, and we are discussing about the artificial intelligence, intelligence powered donor matching, what it is and how it works and why it matters. I'll make the topic into three sections. As I said, what it is and how it works and why is it necessary. So AI powered donor matching, what it is, the NMDP is using artificial intelligence to revolutionize how they find perfect match between patients with leukemia and potential donors. As we were talking, how hard it is to find the donors. It is of 20% chances to find a donor for leukemia. So it is traditionally using the methods is very hard to find the donors, and matching involves complex human leukocyte antigen data, essentially unique genetic markers that need to align between donor and the patient for successful transplant. And using AI, we can analyze the vast amount of data much faster with the greater precision and manual method than the manual methods. And why is it necessary, you may ask? This means finding suitable donors for leukemia patients becomes quicker and more accurate. This speed and precision not only benefit the patients by accelerating their path to transplant, but also significantly reduce the demanding workload on transplant center staff and physicians, allowing them to focus more on the patient care and reduced intensity conditioning. And this is the patients are subjected to chemotherapy and radiation to prepare their body for new cells that are introduced from the donor. So the reduced intensity condition is a newer approach that uses lower, less harsh doses of these treatments, uh, and and this will help the children and teenagers with chronic myeloid leukemia showed that the specific RIC regimen was both safe and effective. By reducing the intensity of conditioning, this approach makes transplants a viable and safer option for more patients, especially for those who might not strong enough to withstand traditional or higher dose regimens, which happens in our daily lives. Some people cannot take or withstand the higher doses of chemotherapy and radiation. And this will help us to broaden the pool of patients who can potentially benefit from a life-saving treatment. The next slide. So Future of stem cell therapy for leukemia, the CRISPR edited stem cells. In this is an exciting area involving using CRISPR or Cas9, often described as molecular scissors or gene editing tool, to precisely correct the genetic mutations that cause leukemia. And this works by taking the hematopoietic stem cells, which are blood forming cells, and using CRISPR to fix the faulty genes in them. Like Dr. Parmar was saying, to fix the faulty genes. And why is it necessary? Is preclinical models show great promise. If successful in humans, this could offer a way to treat leukemia by fixing the root genetic cause in patients' own cells, potentially leading to a cure without needing a doctor. And leukemic stem cell targeting. Why is in there is a major challenge in this treatment. The major challenge is the relapse of the cancer. This often happens because leukemic stem cells survive treatment. There are small populations of highly resistant cancer cells that can regenerate the entire tumor. And the therapies under development are specifically designed to find and eliminate these stubborn LSC cells, which are leukemic stem cells. And by selectively eradicating leukemic stem cells, these new treatments aim to prevent the cancer from recurring, offering a more durable remission and long-term survival of patients. Stem cell-derived immunotherapy. In this therapy, the body uses its own immune system to fight the cancer. This approach takes it a step further by using immune cells like natural cells or neutral cells and the T cells that are derived from the stem cells. How it works. These stem cells derived immune cells are engineered in the lab to enhance their ability to recognize and destroy leukemia cells. And this innovation seeks to create more potent and precise immune response against leukemia, potentially offering a powerful new therapeutic avenue, especially for patients who don't respond to conventional treatments. The next slide. So in this slide, with the stem cell therapy greatly increases survival rates to individuals with leukemia. This point re-emphasizes the tremendous life-saving potential of stem cells transplanting and underscoring their critical role in modern leukemia care. It highlights the direct and profound benefits of patients. And the compatibility is one of the biggest challenges, but is constantly being improved. This acknowledges a significant hurdle in transplantation, how hard it is to find the donor or compatible donor. However, it immediately follows up with a reassuring message of ongoing progress and innovation in matching techniques like AI-powered matching methods, as we discussed before, indicating that this challenge is being actively addressed and overcome. Future technology is continuing to improve and innovate new ways to utilize stem cells to increase survival and remission odds. This looks forward with optimism. It reiterates that the field is not stagnant but is continuously evolving with new research and technological breakthroughs, promising even better outcomes for patients in terms of survival and the chance of remaining cancer. And we would like to thank you for listening and being part of this. We'd happy to take any questions.
Thank you, Kaya and group. Thank you so much, Achita, Kaya, Chris, and Sandeep. There is a question here from Julie. Julie is asking, would the stem cell therapy be used through the bone marrow instead of contacting blood cells directly?
I can answer that one. So as far as stem cell therapy being used with bone marrow, the bone marrow itself is the stem cells. With the way that blood is created in our body, it is for the most part, there are some stem cells directly in the blood, but the majority of blood cells are being created by the bone marrow. And so the main way for stem cells to be utilized would be through that bone marrow and repopulating it. And that way I guess you could say we aren't directly messing with the blood cells themselves, but we're indirectly controlling them that way, if that makes sense.
She has a follow-up question, Kai or anyone. What does chemotherapy do to prepare the patient for stem cell therapy. Chemo typically lowers immune system, so it's a bit confusing.
And I will be answering that. The donor cells are considered as foreign cells or the invaders. So the immune system will be, as you said, the immune system will be lowered to promote the donor cells to settle in the body of the leukemic the patient.
And third question is, would they do blood transfusion to remove the damaged blood cells?
I think I can answer that one. So, using a blood transfusion to remove damaged blood cells would be kind of an ongoing battle, essentially, because it's a recirculating thing. It could be concentrated in a certain region for cancers themselves. However, in terms of removing the actual itself, the blood transfusion, it's flushing it out, removing it would not be that simple, unfortunately.
I can also add additional insight to that question specifically. So with our blood cells are pretty, I don't remember the exact amount, but we're pretty consistently going through our blood. Our kidneys filter our blood pretty often, and so we get new blood cells fairly quickly. And so that's why the focus is on the bone marrow fixing it, because the cancerous cells are in the blood, yes, but the source of those cancerous cells are the cancerous cells in the bone marrow themselves. So if we replace the bone marrow, you might have the cancerous cells in your blood for a while, but they'll be quickly washed out within, I think, a few weeks, just because of how our blood circulates and gets like cleaned through, gets repopulated fairly quickly.
Thank you so much. I think those are the questions for your group. So thank you so much, all four of you, for a very insightful presentation, a very promising area for stem cells to really address leukemia, which is, fairly high up on the list of cancers. All right. Could I please request you to kind of, take, yeah. And then the next group is Rajvi and company. Please upload your presentation.
Yes, I will start sharing my screen and I will go ahead with this.
So remember, keep it to 18 minutes, Rajvi.
Yes. Here we go.
And Rajbi, if you can introduce yourself and the others as to where you'll be doing your internship really quickly.
Hello everyone, my name is Rajvi Parekh. I am a student in the MS Biotechnology and Bioinformatics program. I will be pursuing an internship at Dr. Lam's lab in UC Davis, where we'll be working on drug delivery and proteomic-based techniques, inculcating stem cells as well as cancer cell treatments. Currently, this presentation is going to be based on the potential of stem cells as an alternative therapy in liver cirrhosis. And my colleagues are going to start the presentation.
Hello everyone, myself Hari. I'm doing my internship at UC San Diego under Dr. Jogleson. So, what our project is based on the neurodegenerative diseases. So, as we all know that the neurodegenerative diseases are like pretty complicated to treat. So, we are trying to hunt down the any potential biomarkers using the stem cells and in the in vitro conditions. So if we know the potential biomarkers, then we can create a better and efficient precision medicine, so we can treat the patients in a good way, and so we can increase the quality of life too. So I will just give a quick outline for our presentation. So we will be dealing with the liver cirrhosis. Basically, the introduction about the stem cell, which Dr. Parmar has explained us very well. What's liver cirrhosis is, what are the risk factors, symptoms, and what are the current treatments that are available in the market, and what stem cell can do, as how it can play a major role in treating the liver cirrhosis. What will be the current clinical trials, and what can be the future outcomes and challenges. Next slide, please. Next slide. Yeah, so why we choose stem cells, as they have the four unique hallmark features that make themselves so unique, that is self-renewal, the potency and the ability to differentiate into different kind of cell, and even the regenerative potential of the stem cell that the normal cells lack. So that's the reason this plays a unique role in in the medical field. Next slide, please. So in this slide, you can see I have highlighted two articles. So the first article is application of stem cell in regenerative medicine. So in this article, the author is trying to explain like how various kinds of stem cells, like human induced stem cells, even the mesenchymal stem cells, so they can be used in various diseases like neurodegeneration, liver diseases, cardiovascular. So while talking especially about the liver diseases, this is there are like many clinical trials going on in this field, like for getting the more promising therapies out in the market. So the mesenchymal stem cells are the one which is a potential candidate which we can use for treating the liver diseases, improving the functions. And the next article is stem cell based therapy for humans. So in this, they are majorly focused just on the mesenchymal stem cells, so how we can potentially use them as a treatment options for the liver diseases. As you know, the mesenchymal stem cells can be used for the organ directed therapies, so we can just exactly target where we want to heal, where we want to deliver the stem cells. So this article is based on that. Next slide, please. So the basics about the liver cirrhosis. So what is liver cirrhosis? So let's break down the word liver and the cirrhosis. Means the liver is having a fault. Cirrhosis means a scar on the liver. So next slide, please. Oh, so this is a complicated picture, but yeah, let's just break down into simple terms. So what happens is there are like many factors which causes the liver cirrhosis, such as hepatitis, alcohol. If a person is like more addicted to the alcohol, that may cause liver cirrhosis, autoimmune diseases, and especially there are many cases where the drug induced liver cirrhosis is occurring. So what happens is when these toxins attack the hepatocytes. So hepatocytes are basically the cells which liver is made up of. So the liver is trying to repair itself. So, but there is a failure occurring, and that failure lead to the accumulation of scars on the liver cirrhosis. Sorry, on the liver. So that makes the organization and the orientation inside the liver like in a impaired manner that affects the liver very badly. So in liver cirrhosis, in a way you can define as the healthy hepatocyte cells are replaced by a dead cells by a fibrotic tissue. And next slide, please. So these are the basic causes of liver cirrhosis, as I recently mentioned: alcoholism and drug induced liver cirrhosis, and there may be like immunological causes like autoimmune diseases. They're having a huge ratio. Next is about, oh yeah, next slide, please. So this is about the clinical presentations, means in simple words, the symptoms, what a person can experience if he's having a liver cirrhosis. So as we all know that liver is a potential organ for the deamination and detoxification of the body, as liver clears all the toxins from our blood, right? So what happens if our liver get damaged? So there is a chances of getting a portal hypertension, that is a high level of blood pressure in the veins, and that may causes damage to the spleen. It may causes ascites, that is the swelling of our belly region. And if the liver is damaged, then the most important protein of our body, albumin, that may be lacking, and that may cause swelling to our body. Even the clotting proteins in the liver, so if that's damaged, then there may be a bleeding risk. And even the liver is damaged, then there won't be any purification of the blood, so the ammonia which is in the blood will be getting accumulated, to be very specific, urea. So that may cause encephalopathy too in the later stages, and the most common one will be the jaundice. Next slide. So here are certain complications in the liver cirrhosis, as I already mentioned. So you can see the image which is towards the right hand side of the presentation that clearly indicates like what happens step by steps. So when the liver is damaged, what happens? It starts with a portal hypertension, and it will be leading to the ascites, then to the spleen, splenomegaly, which we consider as, then jaundice, and later to the last two stages that which to the brain, that is the metabolic disorders like encephalopathies and etc. Next slide. So from here, my colleague will continue.
Hello everyone, my name is Debalina and I'll be doing stem cell research under Dr. Laura Perrin at Children's Hospital LA, and we'll be working on use of stem cell on kidney diseases and childhood kidney cancer. So, as my teammate said, cirrhosis is a late stage liver disease in which healthy liver cells are replaced by scar tissues. This disease progresses very slowly, but it can lead to total liver failure. So, liver cirrhosis is divided into two types based on the symptoms. They are compensated cirrhosis and decompensated cirrhosis. So, compensated cirrhosis is the early stage cirrhosis in which in the beginning the body's healthy cells try to compensate for the reduction of liver function when scar tissues accumulated in the liver. So healthy cells try to compensate that reduction of liver function, so the symptoms are usually not visible or very prominent at this stage. This is called early stage compensated cirrhosis, and the late stage but eventually after few more years the symptoms are usually started to become more prominent, and this is called decompensated liver cirrhosis, which is very very dangerous and it can be very fatal. So the symptoms of cirrhosis include weight loss, nausea, vomiting, unreasonable bruises on the skin, nasal bleeding, pain in the right hypochondrium, that is the upper side of the chest where liver exists. Then constipation and diarrhea, loss of appetite, dark urine, and yellowing of the skin and sclera. Next, please. Then the diagnosis of liver disease. Next slide, please.
I am sorry for the delay. There is some issue with the internet. There we go. Sorry about that.
Thank you, no problem. So the doctor usually start with physical exam about symptom, medical history, medication, and lifestyle. They took the signs of liver damage and past liver disease history. So the typical usually the test involve blood test, imaging tools like ultrasound, CT scan, and MRIs are being done, and elastography means liver stiffness are being tested, and that means they check the fibrosis level or scar tissue level in the liver, and then liver biopsy is done. A small sample of liver tissue is taken with a needle, and then it is tested for liver cancer. Next. So the risk factor associated with liver cirrhosis. So usually alcohol consumption is the most prominent common cause of liver cirrhosis, and hepatitis B and C infection also a major contributor of liver cirrhosis. The viral infection causes inflammation inside the liver, and which causes scars in the liver tissue. And another is non-alcoholic fatty liver disease, autoimmune disease where body's own immune cells attack the hepatocytes, and long-term medication use. Certain medicines like methotrexate, isoniazid can cause liver damage. Some inherited conditions like Wilson disease and hematochromatosis also increases cirrhosis. Exposure to toxins like various industrial chemicals and HIV co-infection with hepatitis B and C can cause liver damage, and poorly controlled diabetes, malnutrition, rapid weight loss, and family history increases the vulnerability of this disease. Next. So prognosis of liver disease. So cirrhosis cause prominent liver scarring that doesn't fully heal, but its progression can be slowed and inflammation is controlled if proper treatment and lifestyle changes are are considered. So compensated cirrhosis can allow patient to live around. The compensated cirrhosis means the early stage cirrhosis, which with treatment it can allow patients to live 15 plus year with proper care. But decompensated cirrhosis, like the in case of decompensated cirrhosis, the life expectancy is significantly lower, about 7 years or less. Lifelong liver care is essential to prevent worsening and manage of complications. Prognosis tools like CTP and MELD scores usually calculated for transplant for the to consider the priority transplant list, and severe untreated cases may reduce the life expectancy to under 2 years without liver transplant. So next, current treatment strategies for liver cirrhosis and why stem cell based therapy is needed for this disease. It will be discussed by my team member. Thank you.
Hello everyone, my name is Nirali. So today I will drive you through the current treatment. So what are the current options for the treatment for the liver cirrhosis? So as all of my team had mentioned, the liver cirrhosis is a progressive, irreversible condition marked by extensive scarring. So therefore, the liver cirrhosis cannot be cured, but treatment can help to slow and halt its progression and also to the prevent complications. So basically there are the three treatment options. So first is the lifestyle modification, pharmacological treatment, and liver transplantation. Next slide, please. So this is the treatment algorithm for the liver cirrhosis, where from the early identification to its diagnosis to its treatment and everything is shown. I'll elaborate everything in the next slide. Next slide, please. So first is the lifestyle modification. So lifestyle changes are the first and the most essential step in managing cirrhosis, especially in the early stages. So what are the lifestyle modifications? So avoid alcohol, healthy diet, sodium restriction, regular monitoring, vaccination, and exercise. Next slide, please. Second is the pharmacological treatment. So medications plays a key role in managing cirrhosis to prevent its progression. They help to treat the root cause and control the symptoms and complications. So firstly, antiviral drugs are used when the cirrhosis is caused by hepatitis B or C. Diuretics like spironolactone and furosemide helps to reduce swelling and fluid buildup in the belly and legs. Beta blocker lower the pressure in the liver blood vessels and helps to prevent the serious bleeding from the veins and esophagus. Antibiotics are used to prevent or treat the infections like spontaneous bacterial peritonitis, which is common in the people with the fluid buildup. And the vitamin supplements, especially the vitamin B supplements and the fat soluble ones, are important since the damaged liver cannot absorb them well. Next slide, please. So liver transplantation. Liver transplantation is the only curative treatment for the patient with the end-stage liver cirrhosis. So donors can be from the deceased or a living donor, and the carefully matching is done to reduce the rejection risk. And after the transplant, the patient will need to have a lifelong medication to suppress the immune system and prevent the body from attacking the new liver. Next slide, please. So does all treatment option lead to think about the need of an alternative therapy? So what are the limitations of current treatment strategies? Next slide, please. So they only provide a symptomatic relief. It's not a curative, complex surgical procedure, and also the complications after transplantation, shortage of donor organs, lifelong immunosuppressive therapy, and there are the high chances of rejection after liver transplantation. So here comes a stem cell, and stem cell provides us a promising approach in treating the patient with the liver cirrhosis, especially with the end stage. So stem cell provides us an alternative approach which aims to restore, replace, regenerate the damaged tissue of liver. Along with that, it provides us with the many additional advantages, like it provides a tailored therapeutic approach, slow the fibrosis progression, replace the damaged hepatocytes, and promotes the tissue repair. From here onwards, my colleague will brief you through what are the current stem cell-based treatment plan.
Hello again, everyone. So we emphasized on the severity and the seriousness of liver cirrhosis as such a pertinent disease worldwide, which is why we encourage people to look into stem cell-based therapies and to gain education about the same, which is why I'll be now focusing on a stem cell-based treatment plan. So one of the most commonly derived stem cells for the treatment of cirrhosis has been mesenchymal stromal cells or mesenchymal stem cells, which are easily obtained from human beings, say from the bone marrow or from the umbilical cord after deliveries. So they are pluripotent and easily accessible, as mentioned here. And several studies have shown that infusion of bone marrow derived stem cells can reduce liver fibrosis area. And one of these studies was as back as 2005. So there is ample potential in this field that is just waiting for the right push for application. These stem cells also provide a potential treatment for autoimmune diseases like lupus, rheumatoid arthritis, autoimmune liver diseases, and so on. This especially because MSCs have shown to have anti-inflammatory properties as well. And while they are like every other cell, they can be immunogenic, but they tend to show more promise than other cells. I'll go to the next slide real quick. So there are, as of 2024, there are 73 clinical studies involving MSCs for treating liver disorders that have been registered on the NIH clinical trial database. As of this year, there are around 10 or so studies that are currently ongoing or have like unknown status. As of now, they are based all around the world, and they are currently focusing mainly on mesenchymal stem cells in the form of injections or in the form of like specific deliveries to the liver in order to see how what potential do they have and how well they can work. So primary focus is on end stage liver diseases or liver cirrhosis that involve 30. There were 31 trials, and liver failure or decompensation, that are 34 trials. There are fewer trials targeting other liver diseases, including transplantation complications, and single trials for autoimmune hepatitis, Wilson's disease, and biliary atresia. So now, considering we have learned all of this, now we are wondering, so what's next? Why aren't we using this already? Why is liver transplant still one of the last ditch attempts in treating patients with cirrhosis? So the issue is that people with cancer and alcohol use disorder are unfortunately not very suitable for transplantation because of the entire damage in their organs. And transplant candidates have the risk of facing complications like rejection, infection, risk of cancer, and so on. Stem cells that are derived from bone stem cells, like I mentioned before, are easily accessible, especially the mesenchymal stem cells. Apart from mesenchymal stem cells, when macrophages have potential, and macrophages can be isolated from blood because they are white blood cells. So these can be isolated easily and are accessible, and even from, say, adipose tissue. So immunogenicity is a concern when we are isolating these cells from, say, even from the same donor in case of autoimmune or from different human donors. They may elicit variable immune responses that result in adverse outcomes. So understanding the detailed mechanisms of MSC is limited, and they are hindering the treatment protocols and patient selection criteria. Which brings me to the point which Dr. Parmar and another participant brought it up before, that there are several private clinics that claim that they can cure liver disease and related issues by just giving stem cell injections, which are isolated either from the patient itself or from a separate donor. Most doctors, most scientists will not agree or recommend or encourage these type of treatments, because while there are so many trials going on, while there are so many studies going on, the FDA has not approved stem cell therapy just for liver disease. They have approved it for hematopoietic diseases, but not for liver diseases. And a lot of clinics that are based outside of the USA or like work on fringe loopholes claim that they can cure liver disease just by injecting stem cells. So this is a very dangerous situation, and it teeters, I don't want to use that word, but it goes on potential fraudulent levels. So
Rajvi, we'll have to end here because the other
all
group will not get time. I think we should.
that's okay. Yeah, this was the last slide.
Yeah, please.
So
I don't want the other group to get
no
left.
worries. This was my
Okay.
last slide. So
Thank
if you
you.
have
Thank
any
you.
questions, you may please let us know privately, and I'll just end the presentation here.
Loves. Thanks. Thank you so much. Great job. Jaini, can you please upload your presentation?
Hi, my name is Jeannie. I will be completing my internship at Stanford University under the supervision of Dr. Sean Wu, and we're looking into stem cell therapies and 3D bioprinting applications for certain cardiovascular diseases. My team members also will now introduce themselves and where they're going.
Hello, everyone. My name is Dave Atienza, and I'll be doing my internship at USC under the guidance of Dr. Lee, and I'll be working on developing a kidney from stem cells.
My name is Wen. I'll be at USC as well under Dr. Ishida. Our research focus is on understanding how genetic and environmental factors contribute to neurodegenerative diseases.
Hi, my name is Neila Wadja, and I'll be doing my internship at UCSB under the supervision of Dr. Dennis Clegg, and I'll be working on how stem cells differentiate into ocular cells. Thank you.
Perfect. So our team will be presenting on stem cell-based therapeutics for congenital heart disease. So just as an introduction, congenital heart disease, or CHD, this refers to structural abnormalities of the heart or the great vessels that are present at birth. It's actually one of the most common major birth defects, and it affects about 1 in every 100 live births globally, and it accounts for nearly 1/3 of all congenital abnormalities. In 2021 alone, there was an estimated of 4.18 million children under the age of 5 that were living with CHD worldwide. It's also a leading cause of death from congenital abnormalities, responsible for about 4.2% of all neonatal deaths. In low resource settings, CHD is actually rapidly becoming a top cause of under 5 mortality. And even in high income countries where surgical techniques have advanced significantly, CHD still remains a lifelong disease. Patients often require multiple interventions, and they still face a risk of heart failure, arrhythmias, and the need for eventual transplantation. Current treatments for complex CHD, for example, like HLHS, they typically involve staged surgical approach. And this includes the Norwood, Glenn, and Fontan procedures. And these surgeries are life-saving, but they're not particularly curative. One major limitation of this is that surgeries don't regenerate myocardium. It simply reroutes blood flow. So underdeveloped ventricles still remain functionally weak, and long-term heart failure is still a significant risk. Surgical scarring is another issue. This disrupts the heart's electrical rhythm, and it increases the likelihood of arrhythmias, especially in patients with repaired tetralogy of Fallot. We also rely on prosthetic materials that don't grow within the child, and these can calcify or wear out over time, and this leads to repeated surgeries. Even the best surgical outcomes often involve a lifetime of follow-up procedures, complications, and eventually, for some patients, the need for a transplant. And this is where stem cell therapy comes in. As mentioned by Dr. Parmar earlier, stem cells are undifferentiated cells that can self-renew and differentiate into specialized types, including cardiac cells. So we're looking at 4 major types here: embryonic stem cells, iPSCs, mesenchymal stem cells, and cardiac progenitor cells. And these are already somewhat pre-programmed for heart repair. Each type of these stem cells has its own benefits and limitations, like we've heard earlier, and we're learning how to apply them to CHD in a way that's both safe and effective. So stem cell therapy is especially promising in CHD for multiple reasons. The first being pediatric patients have more regenerative potential than adult hearts. And this gives us a better chance of simulating meaningful growth, especially in underdeveloped structures like the hypoplastic ventricle. Second, surgery doesn't restore lost muscles or vessels. It just reroutes circulation. So stem cells can help kind of fill that gap by promoting endogenous repair. Third, children's immune systems are more tolerant and their tissue is still growing and remodeling. And this makes cell engraftment more effective. And finally, stem cells release pro-regenerative signals that promote blood vessel formation, reduce fibrosis, and help existing cardiomyocytes survive. This opens the door to improving long-term heart function after surgical repairs. So stem cells support heart repair through 5 key mechanisms. They secrete growth factors like VEGF, IGF-1, and HGF. This is called paracrine signaling, and this is to promote healing and reshape the heart's microenvironment. They can be pre-differentiated into cardiomyocytes or endothelial cells and directly integrated into the heart muscle, especially when using iPSCs or ESCs. They also modulate the immune response. For instance, mesenchymal cells, they secrete IL-10 and TGF-beta, which reduce post-surgical inflammation and scarring. They stimulate angiogenesis through factors like VEGF and PDGF, increasing blood supply to damaged areas. And finally, they reduce apoptosis and support favorable modeling. preserving cardiac function and minimizing scar tissue formation results from this. So altogether, these actions allow stem cells to kind of act as a biochemical repair system for our heart.
I'll be going different cell types used in therapeutics for congenital heart disease. The first is induced pluripotent stem cells, or iPSC. They are adult somatic cells that have been reprogrammed back into a pluripotent state, inducing, this means that they can self-renew and become any human cell type. iPSCs can be derived from patients' own cells and used to model their specific disease. So for example, researchers can replicate genetic or acquired cardiac disorders in vitro, which they can study in detail without any invasive procedures. In therapy, iPSCs allow us to test the patient's cell's response to a specific drug before actual treatment. iPSC can also be engineered into 3D heart tissues from patients, and it reduces the risk of immune rejection. The second one is mesenchymal stem cells, or MSCs. They are multipotent stromal cells capable of both self-renewal and differentiating into various tissue types. They play a vital role in cardiac repair by secreting growth factors, modulating immune responses, and promoting tissue regeneration. In terms of safety, clinical trials such as TCAP and Perseus have demonstrated that MSC therapies are well tolerated in children with congenital heart disease. Another key example is the Perseus trial, which found that repeated MSC administration significantly improved cardiac performance in pediatric patients with severe heart failures. Lastly, the cardiac progenitor cells, or CPCs, are multipotent cells found in developing heart. They have the ability to differentiate into cardiac lineages, including cardiomyocytes and smooth muscle cells. CPCs are typically isolated from fetal or neonatal heart tissue using enzymatic digestion method. This extraction preserves their regenerative capacity. To enhance their differentiation, CPC can be treated with agents like 5-acetidine, which promotes the transformation into cardiomyocytes. A key example of this is the Apollon study, which demonstrated the CPCs can successfully differentiate into and contribute to cardiac repair.
So here we're going to highlight 3 recent or ongoing clinical trials that are applying stem cells for the treatment of congenital heart disease, specifically a condition called hypoplastic left heart syndrome. Hypoplastic left heart syndrome is essentially a birth defect where the left side of the heart does not develop properly and is therefore unable to pump blood through the body. As mentioned previously, the standard treatment for hypoplastic left heart syndrome currently involves reconstructing the heart using the Norwood, Glenn, and Fontaine procedures to allow the right ventricle to basically supply the blood to the entire body. In these cases, heart failure is caused usually by the overexertion of the right side of the heart as it tries to compensate for the left side. Currently, there are no available treatments that recreate the entire or the part of the heart for transplant. So we are going to look at some stem cell therapies that might help. Our first trial is the ELPIS trial, which uses immune-matched donated bone marrow mesenchymal stem cells to try to strengthen the right ventricle of the heart to prevent or delay the heart failure. These mesenchymal stem cells are injected into the right ventricle of the heart after the second of the 3-step heart reconstruction surgery. For the trial, 10 patients were enrolled and treated in the phase 1 clinical trial. The primary goal of phase 1 clinical trials is to determine first the safety, and then we'll look at whether or not the treatment is effective. Of the 10 patients treated, none experienced major adverse cardiac events, and all are alive and transplant-free after 1 year. They found that the cardiac structure and the function of the right ventricle were also preserved. The researchers suggest that the benefits might be from the exosomes that are excreted by the mesenchymal stem cells, which carry regulatory RNAs that might help with the regeneration and cell survival of the cardiomyocytes. A phase 2 ELPIS trial is currently ongoing to continue exploring the use of these mesenchymal stem cells. The second trial is coded NCT 01883076. It uses a similar strategy of injecting stem cells into the right ventricle during the second stage of the three-step palliative surgery. The cell choice here, though, is we're using autologous umbilical cord derived stem cells, as Dr. Parmar has taught us earlier. The primary results from the study were similar, with no significant safety concerns and a preserved right ventricular function and normal heart growth rate. I'll talk a little bit more about this trial in the next slide. But the third trial is the TCAP trial, where they use patient-derived cardiospheres, which are cardioprogenitor cells. They were isolated from heart tissue that were removed during the first of the 3 palliative surgeries. Similarly, the cells were injected into the coronary artery during the second stage of the 3-step surgery, and the results show that there were no safety concerns and that patients had improved right ventricular function at 3 months follow-up. So looking back at the second of the 3 clinical trials, so patients were diagnosed with hypoplastic left heart syndrome as infants, and they go through the first stage of the reconstruction surgery at the age of 4 days. The right ventricular ejection fraction, which measures how well the heart ejects blood, was around 35%, which is considered moderately dysfunctional. BNP, B-type natriuretic peptide, an indicator of heart failure, was severely elevated. At the second stage of reconstruction surgery, the patient's cord blood this time, stem cells were injected into heart muscles of the right ventricle. At 3 months follow-up, the right ventricular ejection fraction was increased to 50%, which indicates almost near normal function. And the BNP level dropped all the way down to around 1,000 picograms per ml, which is around 24-fold decrease. The stem cell treatment was found to be safe and effective, which is really promising.
So now that we've got a clear understanding of congenital heart disease and why stem cell therapy is such a promising direction, I'm going to talk about how these stem cells are delivered to the heart. Here are 3 main delivery methods. First, we have the intracoronary injections method, which delivers stem cells into the coronary arteries through a catheter. The idea is that the cells will travel with the blood flow and reach the underdeveloped areas. It's less invasive, but this method it's not always precise for targeting specific areas. The second approach is myocardial injection. This one involves injecting stem cells directly into the heart muscles using a fine needle. It's often done during surgery, and the big advantage here is precision. The third method is a patch or scaffold delivery that uses a biodegradable patch seeded with stem cells, which is placed on the surface of the heart. It supports the cells and helps guide tissue repair and growth. Despite the potential, stem cells therapy comes with some serious challenges that researchers are still working on. I'm not going to go over the details because of lack of time. But if you have any questions, please feel free to ask. So the challenges are the arrhythmias, which is caused by transplant stem cells that may disrupt the heart's electrical signals. We also have the immune rejection, the tumor risk, and another risk is the short lifespan. Here we have the future directions, which is like new ideas to improve stem cell therapy. We have the 3D bioprinted cardiac patches that uses the patient's own stem cells. We also have the personalized regenerative therapy. And one of the most important is CRISPR gene editing. So, what we should take away from this is while CHD is a lifelong condition, the emerging field of stem cells therapy offers a real regenerative hope. Early clinical trials are showing encouraging results in terms of safety and efficacy, which could become a transformative options for patients. However, for stem cell therapies to become viable long-term solutions, we must address challenges related to long-term outcomes, delivery mechanisms, and ethical consideration. Thank you so much for your attention.
Thank you so much, Jenny and company. And I think we're very close to the end of the time here. It's almost 3 p.m. Let me quickly read really quick here. I guess there's no questions. It's just a lot of word of kindness and gratitude. Thank you so much, all these students. It was great having you. I know we're at 3, so we cannot go forward. But if you do have any questions, please do not hesitate to email me, and I can send those questions to the students. And I'm getting responses like brilliant young students. Thank you so much. Yes, these are our brilliant young students, and I have very high expectations from them. They will probably contribute to the workforce, which is going to bring cutting edge therapies, in the next decade or so. So the effort of each one of these students is going to make a big difference in what kind of therapies are going to come to the market. So thank you so much and have a great rest of the week. And thank you guys. Great job, all of you.
Yes,
Thank you.
thank you all so
everyone.
much
Bye-bye.
for your
Thank you, everybody.
knowledge and expertise.
Thank
We
you, everyone.
appreciate it. All right. Thank you.
Thanks, Shana.
Thanks, Shana.